Welcome to the Deep Dive. This is where we take all your sources, your research, technical docs, and, well, we boil it all down to what you really need to know. It's your shortcut to getting properly informed. Today, our mission is pretty fundamental. We're diving deep into the cell. If you want to get beyond, the basic school description and really understand the architecture of life's basic unit, you're in the right place. So we know the cell is the basic structural, functional bit of the body. And it's the simplest thing we actually consider living, right? Like a bacterium or, well, one of the trillions making up you.
And that scale is it's immense. You mentioned trillions of cells in the human body, but the specialization is key. Your sources mentioned something like 250 different types of cells.
250, wow.
Yeah, each one fine-tuned for a specific job. Think nerve cells, muscle cells. Totally different jobs, totally different setups.
Okay, so it's not just a basic block, it's a huge organized community.
Yeah.
To really get how this tiny system with trillions of members works so well, we need to look at the structure. Let's start at the edge.
Yeah.
The boundary.
Right. Let's begin with the plasma membrane. People often call it the cell's gatekeeper. But it's not just a wall, it's dynamic. Fluid.
Dynamic and fluid, I like that.
Yeah.
It's just movement, change, not just holding things in. So what's it actually made of? What gives it that quality?
It really comes down to three main things: lipids, proteins, and some carbohydrates. The core structure is what's called the phospholipid bilayer.
Okay.
Phospholipids, they have a head that likes water hydrophilic and two tails that hate water hydrophobic. So when you put them near water, like inside and outside the cell, they naturally line up. They form this double layer with the tails pointing inwards, shielded from the water. That forms the basic barrier.
But just a barrier isn't enough.
Right.
It needs stability.
Yeah.
And ways to get things across.
Exactly. A pure phospholipid bilayer wouldn't be stable enough, especially with temperature changes. That's where cholesterol fits in. It wedges itself between the phospholipids. Think of it as like temperature regulation for the membrane itself. It stops it getting too rigid when it's cold or too floppy when it's hot. Keeps things just right.
Maintains the integrity.
Okay. But how does the cell control what gets in and out? That selective permeability thing.
That's where the proteins come in. They're embedded within this fluid layer, like icebergs floating in the lipid sea. They act as channels, pumps, receptors, basically the border control.
So they actively choose what passes.
Very actively. They let in nutrients, ions, signals the cell needs, and they keep out unwanted stuff or harmful things. The membrane is constantly managing traffic.
Okay, got it. So let's move inside now, past that busy border. You cross the plasma membrane, and you're in the main body of the cell, the cytoplasm. Sources describe it as a thick solution. Sounds a bit simple.
It does sound simple, but it's a whole internal environment, and we should be a bit precise here. Cytoplasm technically means everything inside the membrane except the nucleus.
Okay.
The actual fluid, the jelly-like stuff itself, that's called the cytosol. It's mostly water, but it's thick with salts, proteins, all sorts of molecules. Lots of chemical reactions happen right there in the cytosol. And within that cytosol, you've got, well, not just floating, but structured components. Before we hit the organelles, though, there's the scaffolding, right? The cytoskeleton. Already gives stability.
It definitely provides stability, yeah, but stability almost undersells it. If the cell's a city, the cytoskeleton isn't just the building foundations, it's the roads, the monorails, the internal transport network.
Okay, now we're getting to the really interesting parts, the command center. Let's talk about the nucleus. It holds the blueprint, right? What's its main job, its core structure?
The core function is, well, protection and control. The nucleus houses all the DNA the instructions needed to regulate everything the cell does. It's the master library. And protecting that library starts with the nuclear envelope. It's actually a double membrane, 2 layers thick. And crucially, it's dotted with things called nuclear pores.
Pores, so things can get in and out.
Yes, but in a very controlled way. These aren't just simple holes. They're complex protein structures that act like security checkpoints. They let essential messages like RNA out and specific proteins in, but the DNA itself stays safely inside.
Okay, so the DNA is secure inside. How is it organized in there? We hear about chromatin and chromosomes. Are they different things?
They're different forms of the same thing, really. Most of the time, the DNA isn't just a loose strand. It's wrapped around proteins, specifically proteins called histones. This DNA protein complex is called chromatin. Think of chromatin as the working form of DNA. It's somewhat uncoiled, accessible, so the cell can read the genes it needs.
Right.
But when the cell needs to divide, copying all that DNA and splitting it perfectly is a huge task. So the chromatin condenses. It coils up incredibly tightly into those dense rod-like structures we recognize as chromosomes.
So chromosomes are just tightly packed chromatin for moving day, basically.
That's a great way to put it. Makes division manageable.
And inside the nucleus, there's another structure too, right? The nucleolus. What's its specific role?
The nucleolus is a dense spot inside the nucleus. Its main job, its critical function, is making ribosomal RNA, rRNA, and assembling the subunits that will become ribosomes.
Ribosomes, protein factories. We'll get to those.
Exactly. So the nucleolus is basically building the protein-making machinery right there next door to the instruction manual. Very efficient.
That makes perfect sense.
Okay, let's follow the workflow out from the nucleus. We need to talk about the manufacturing and shipping department, the endomembrane system.
Right. And that system starts right next to the nucleus with the endoplasmic reticulum, or ER. It's this huge network of interconnected tubes and flattened sacs. And importantly, its membrane is actually continuous with the outer membrane of the nuclear envelope.
So it's directly connected, a seamless path.
A seamless path. Now, there are two types, visually distinct. The rough ER looks rough because it's covered in ribosomes.
The protein factories we just mentioned.
The very same. So the rough ER's job is mainly synthesizing proteins, especially those that are going to be shipped out of the cell or become part of a membrane or sent to certain organelles like the Golgi. It also helps sold these proteins correctly. Quality control is a big part of it.
Quality control. So if a protein isn't folded right, the rough ER can often detect that and deal with it. Misfolded proteins generally don't get shipped onwards. Then you have the smooth ER, no ribosomes, so it looks smooth.
Different function, then.
Totally different. The smooth ER is key for making lipids like steroids and hormones. It's also crucial for detoxification, breaking down drugs, alcohol, metabolic byproducts. Your liver cells have loads of smooth ER for this reason.
Okay, rough ER for proteins, smooth ER for lipids, and detox. Now, these newly made molecules, they need processing, sorting, packaging.
Right.
That sounds like the Golgi apparatus, the cell's post office.
That's a classic analogy, and it works pretty well. The Golgi is a stack of flattened membrane sacs called cisternae. Think of it like a stack of pita bread, and it has directionality.
Directionality.
Yes. Products, usually arriving in little transport bubbles called vesicles from the ER, enter one side, the cis-face. They then move through the stack, cisternae by cisternae, towards the other side, the trans-face. As they travel through, they get modified. Sugars might be added or tweaked, molecules sorted based on their final destination. They get tagged with molecular signals like zip codes.
Zip codes, okay.
Then at the trans-face, they're packaged into new vesicles, which bud off and carry the finished product to wherever it needs to go, maybe the plasma membrane for release outside the cell or to another organelle.
That's an incredibly organized system.
Yeah.
Manufacturing, quality control, processing, shipping. But any complex system generates waste or has parts that wear out.
Yeah.
What about cleanup and recycling?
Yes. That critical role mainly falls to the lysosomes. These are small sacs bound by a single membrane, and they're filled with powerful digestive enzymes, hydrolytic enzymes.
Digestive enzymes, so they break things down.
Exactly. They can digest food particles taken into the cell or destroy invading bacteria or viruses. And very importantly, they perform autophagy.
Autophagy. I've heard that term. What does it mean at the cell level?
It literally translates to self-eating. It's the cell's internal recycling program. Lysosomes fuse with old, worn-out, or damaged organelles, like an old mitochondrion maybe, and break them down.
And recycle the parts.
Yes. The molecular building blocks are released back into the cytosol to be reused. It's absolutely essential for keeping the cell healthy and removing junk.
Okay, this whole dynamic city building, shipping, cleaning it must take a huge amount of energy, constant energy flow. Let's talk about the power plants.
Absolutely. The primary power generators are the mitochondria. Singular mitochondrion. These are the sites of cellular respiration. They take fuel molecules like glucose and oxygen and convert their chemical energy into ATP adenosine triphosphate.
ATP, the energy currency of the cell.
Precisely. ATP is the readily usable energy packet that powers almost everything else we've discussed, building proteins, pumping ions, moving vesicles along those microtubule tracks. It all costs ATP.
And structurally, mitochondria have two membranes. Why is that important?
That double membrane is key to how they work. The outer membrane is smooth, but the inner membrane is highly folded into these structures called cristae.
Folds. Like shelves.
Kind of. These folds massively increase the surface area inside the mitochondrion, and it's on the surface of this inner membrane that the main ATP-generating reactions happen. More surface area means more space for those reactions, more efficiency. But here's something really fascinating, tying back to what you found in the sources. Mitochondria have their own DNA, a small circular piece of DNA completely separate from the main library in the nucleus.
Own DNA inside the power plant. That is interesting. Semi-autonomous, you said?
Pretty much, they can replicate some of their own components. It's a really intriguing feature.
Okay, definitely putting a pin in that independent DNA idea. Before we wrap up the energy side, what about the actual machines that build things using that ATP? The ribosomes.
Right, the ribosomes. We mentioned them on the rough ER. They are the actual molecular machines that synthesize proteins. They read the messenger RNA instructions sent out from the nucleus.
The blueprints.
Exactly. And they link amino acids together in the specific sequence dictated by that blueprint to build a protein chain. Cells need millions of them because protein synthesis is constant and vital. And we find them in two main places, which relates to where the protein product is needed. We talked about the bound ribosomes stuck to the rough ER.
Making proteins for export or membranes.
Correct. And then there are free ribosomes just floating around in the cytosol. They make proteins that are needed right there within the cytoplasm itself. Things like enzymes for metabolic pathways in the cytosol or the tubulin protein needed to build the cytoskeleton.
Okay. So let's step back. We've gone from the boundary through the internal structure, the control center, the manufacturing and shipping, the cleanup crew, and the power plants. It's an incredible level of organization and integration.
It really is. And the big takeaway, I think, is the importance of compartmentalization. The eukaryotic cell works because it separates different functions into different membrane-bound organelles.
Right.
You have detoxification happening safely in the smooth ER, acidic digestion contained within the lysosomes, energy production concentrated in the mitochondria. This separation prevents interference and allows very different chemical environments and reactions to happen simultaneously within the same tiny cell. That's the genius of it.
It truly is. And that leaves us with a really thought-provoking point, circling back to that unique detail about the mitochondria. We established the nucleus holds most of the DNA, directs basically everything.
Right, the central command.
But we also confirmed that energy production, arguably one of the most critical functions, happens in the mitochondria, which, well, they carry their own separate DNA, their own little instruction booklet.
Independent genetic material.
So, if the nucleus controls all cellular activity, what does it really imply? That the power generation system is partly governed by an organelle with its own protected, separate genetic information? Is that just redundancy? Or does it point to something deeper about control, maybe its evolutionary history, something for you to think about?